right? Any activity, whether you're driving a car or you're going out for a meal, carries a risk with it, right? It's the same thing that's true when you think about therapy. Is what you're trying to do is mitigate the risk so that there is a responsible risk-benefit ratio to any particular therapy, and that's true for whether you're looking at small drugs as well, which are in the clinic, right? That's why you have adverse event reporting. So what the FDA has done is to look at cells, look at the properties, and define what is the risk-benefit ratio depending on the indication you want to use it for. In some cases, it's okay if you have an incidence of 10% of a leukemia, for example, in a gene therapy trial, for example, because the risk of death is near 100% with the absence of that therapy. 10% leukemic indication for something that you're taking for a headache pill would be completely unacceptable, right, in terms of doing that. And so that's the sort of balance that people have. But there are very simple, I wouldn't call them simple, but there are well-tested models for how you develop these sorts of risk-benefit estimates in terms of doing it. And I think the same models could apply for cell therapy as well.
So I think one of the great things is that the FDA and other regulatory agencies recognize that there's a lot of uncharted territory here in terms of how you actually put in place mechanisms and a clear pathway to validate products, characterize those products, test them for safety across a range of different things. I mean, we have challenges related to cell therapy-based products that maybe we don't have when we're dealing with small molecules or traditional biologics. And so you've got to worry about things like ectopic tissue or tumor genicity or other things like that. So creating a framework and doing it in a coordinated way with the regulatory agencies is in everybody's best interest. We need to have clear, transparent standards that tell us what is it that we actually need to do from a preclinical and from a clinical perspective to ensure patient safety. And I think you're right. We've actually been pretty fortunate about the consistent safety pattern that we've seen over the past 10 years and beyond in terms of a broad range of clinical studies that have been run with various types of cell therapies. I personally think there's a reason for that. Okay? I think that with traditional pharmaceuticals or even with synthetic biologics, let's start with pharmaceuticals. You can have a lot of effects like off-target effects or things like that because it's an engineered synthetic molecule that doesn't exist naturally in the body, and there aren't really good models to allow you to assess on a genome-wide way whether or not you're going to see some sort of untoward toxicity or off-target effect. With protein therapeutics, the track record has actually been significantly better from a safety perspective than it's been for traditional pharmaceuticals because proteins are things that tend to exist in the human body. And so, as a result, there's a higher probability that those things are going to be safe. It doesn't mean they're always going to be safe, but it means that there's a higher probability because they exist naturally in the human body. With cells, I think the same argument applies. If you look back over the risk profile, there was a very interesting study that was published early this year that looked at the traditional risk factors and attrition rates in development between pharmaceuticals and biologics. The success rate for biologics is substantially higher than it has been historically for pharmaceuticals. And again, I think this is the reason why. But we shouldn't take the clean safety profile that we've seen to date for granted. The regulatory agencies certainly aren't, and I don't think we as an industry should. So what we need to do is really strive for that transparency in terms of what specifically are the safety tests that we need to do, make sure that information is broadly distributed, whether it's through guidance documents or other things that we can do to create a very clear and transparent landscape. That will allow all of us to use our resources more efficiently, and that's exactly one of the things that ARM is focused on in terms of interacting with the FDA and other regulatory agencies to create that transparency, whether it's around cell potency standards or safety testing or other things, and get there as quickly as possible.
You have time for one more question. Yep, there we go.
So, as a CEO of a hospital that does clinical trials, and given where healthcare costs are in the United States and they're escalating, I think we have a great opportunity to use cellular therapies as an example that will improve health and decrease cost. And so your comments about going to CMS and other regulatory agents simultaneously while we're going to FDA is critical because the time frame between going to get CMS approval, to get reimbursement for a procedure that has good documentation is extensive. And I think we are not talking a lot about efforts about how this is going to actually improve healthcare delivery and decrease costs because of the impact in these chronic illnesses that cellular therapy can have. I'd like to hear your comments on that.
Sure. Well, I'll tell you, the history of the industry, particularly over the past decade, and it has been, people have really shied away from the whole concept of comparative effectiveness, right? Comparative effectiveness can mean a lot of different things. What I think we need to be doing is we need to be putting in place analytical capabilities in the clinical development process that evaluate pharmacoeconomic effectiveness and specifically the cost effectiveness of the therapies that we're trying to develop. We don't live in an era anymore where we can say we delivered a great drug that has a phenomenal impact, but it's hugely expensive, but that's okay. We have to show clinical effectiveness and economic effectiveness. And the way to do that is to actually start building that analytical framework into our clinical trial design. Now, I think that is a big shift in thinking for the entire industry. But in the field of regenerative medicine, it is in our best interest to make sure that we incorporate that kind of mentality as quickly as possible. The beauty of it is that if we do this right, particularly with some of the indications that we're going after that represent enormous clinical cost burdens, that we will actually get to a point where we can deliver therapies that, yes, they may be expensive, but we can illustrate in very specific and quantitative terms how cost effective these therapies are. Our goal is not just to improve clinical outcomes and enhance patient quality of life, it's to shift the cost curve in the right direction. We have to innovate our way to get to that point. That's one of the things that Washington, unfortunately, has not paid enough attention to is how do we foster that type of activity, and that needs to change. And that's one of the things that the Alliance is really focused on.
Susan, what? Go ahead.
Yeah, I think the experience of ARM is probably, I think it was within weeks of being formed, there was an FDA briefing, and I think everything that the Alliance is doing is sort of early and often when it comes to interacting with the regulatory agencies. And I think that we'll be able to make that point about economic effectiveness of even expensive therapies. But I think also what we'll see is, hopefully with the IPS and other technology, by being able to have more accurate sense of toxicity going in to designing a trial, that some of the costs of actually conducting the trials that have in turn driven up the cost of delivering the therapies are going to be able to be controlled.
Well, I tell you, I have personally benefited a lot by your insights here on the panel, all of you. And Mahendra, thank you very much. Matthias, thank you. Susan, thank you very much. And Gil, if we could give our panelists a round of applause for sharing.